Good afternoon and welcome virtually to Old Dominion University and its remote experience for young engineers and scientists program known as REYES. My name is Giovanna Gennard, Assistant Vice President for Strategic Communication and Marketing and co-chair of REYES, and I will serve as your host and moderator for today's session, Tissue Engineering and Health Sciences, featuring Martina Zamboni, Mackenzie Tardif, and Caleb Wyckoff. Our special guests will talk about the focus of their work with the engineering of cells and the use of modified stem cell lines, including the generation of cellular models for the study of neurological diseases. A second focus of their innovative work is the generation of tissue-specific substrates, which allows them to grow and study mammalian cells in an environment similar to that of a human body. This work takes place under the principal investigator, Peter Malika, who is an associate professor in the School of Medical Diagnostics and Translational Services and is the technical supervisor of ODU Monarch Molecular. Throughout the session, you may submit questions through the Q&A box on your Zoom screen. And following the presentation, we will answer as many of your questions as time allows. It is now my pleasure to introduce our guest speakers from Old Dominion University. Would you please turn on your camera so we can see you? Martina Zamboni is a PhD student in the Department of Biomedical Engineering. Her research focuses on the study of stem cell division and differentiation and how these processes are affected by the cellular microenvironment. Mackenzie Tardif is a PhD student in the Department of Biomedical Sciences. Her research focuses on the characterization of Huntington's and Alzheimer's diseases stem cell models, as well as the application of picosecond pulse electric fields as a potential treatment model. Caleb Wyckoff is a PhD student in the Department of Biomedical Sciences. His research focuses on the generation of tissue-specific substrates and their application in the study of cell proliferation and neurological disease. Please join me in welcoming our presenters to REYES. Welcome, everyone.
Thank you so much. Well, I guess we can get started. I'm going to get started sharing my screen. All right. As you just heard, today's presentation is going to focus on tissue engineering and regenerative medicine, and particularly the work that we do here at the Stem Cell Lab at Old Dominion University. I will go through there you go. So having a little bit of technical issues. We will go through first a little bit of background on what tissue engineering and regenerative medicine is, the way that we intend it in this lab, and then we're going to focus on what specifically we do in the laboratory, particularly when it comes to using stem cells, the generation of tissue-specific substrates, and then we're going to give a quick look at the biomedical devices that we use on a daily basis, specifically our 3D bioprinter and our pulsing device. So for what concerns our background into these fields, when I was thinking about this presentation, I was going a little bit back and forth between, tissue engineering and regenerative medicine. What can best define what we do in the lab? But in reality, these two fields are very much intertwined. They rely on each other. And they're very similar in many aspects. So I think these two images that I pulled up here are very representative of what we do. Very briefly, tissue engineering, the way we intend it here is the use of engineering technique, engineering principles to generate tissue grafts and the application of cells as well to generate our tissues in an artificial manner that could potentially either just represent what goes on in the human body and can be used as a model, or that could potentially be applied in the field of medicine. When we talk about regenerative medicine, we take a step further and we do apply many of the principles of tissue engineering, but we also consider the ability of the natural properties of the human body and the self-healing processes that the body has already, and we try and apply the principles of engineering and what we can do artificially in our laboratory, and have them supplement our what our body is naturally able to do to be able to promote regeneration of tissues. I like this first image because it's very representative of both major aspects of our laboratory. We do a lot of work in for the generation of tissues, like it was introduced earlier, tissue-derived substrates, I'm sorry. There's many ways in which we can do this, and this is a very broad category. It goes from applying, making a completely artificial substrate that we can possibly implant in an organism, or we can grow cells into, or it can intend taking donor tissue and decellularizing, so treating it chemically and then using it for further application. Tissue engineering, when it comes to cell models, can again mean a lot of different things. As we will see, even in our lab, it means a lot of different things. We can use it simply to, it can mean simply engineering the cells so that we can see something better, see what goes on in the cell themselves and study the actual cellular processes. Or we can engineer the cells to generate some, for example, disease models that we would not otherwise be able to get naturally. Or we can combine, again, the application of the use of cells, engineered or not, to the use of scaffolds to create tissues artificially. And for what concerns the regenerative medicine aspect of our laboratory is, once again, the application of engineered cells as well as engineered tissue to study neurodegenerative diseases, as well as being able to just create models of disease or non-disease tissues that are a little more representative of what our body actually is like, and not just and kind of moving forward from the traditional lab environment that is not often representative of the true organism's conditions. So the first thing that I will go in a little more depth is the concept of stem cells and what we are interested in looking at. So you might already have heard of this, but what are stem cells? Stem cells are, we define as stem cells all the cells in the body that are able to differentiate into more specialized cells, as well as divide and create new stem cells. So they have this ability to divide into either a new stem cell or a differentiated cell. Now, the number of cells, of types of cells into which one single stem cell can differentiate is a property that we call potency. This word is very important because it is used to define the category into which different stem cells can fall. We identify 4 different types of stem cells, starting from the very earliest one that we encountered during organism development. We have totipotent cells. A totipotent cell is a cell that can divide and form into all of the cells that are necessary for organism development, which means all of the cells that are in the human body can originate from a totipotent cell, as well as the cells that are necessary for support structure of an embryo, for example, the placenta structure. Then we move into pluripotent cells. These are similar to totipotent cells, but they can only form the cells that we find in the human body. So they can differentiate into just about any other cell, but no support structure. Multipotent cells are cells that have a more limited ability to differentiate. It means they can only differentiate to a subcategory of cells. These are also the type of stem cells that we will find in an adult organism. And for example, are the cells that you will have in your that are able to generate all your blood cells. So your bone marrow, you'll have blood stem cells, and these can only divide to the cell type that are found in your blood. Differently, we can have, for example, neural stem cells. There are different types of stem cells, and these can only differentiate into the type of cells that are found in your nervous system. Now, a neural stem cell is already committed to a type of differentiation, so it will not be able to form differentiated cells that are outside of this scope. So, for example, they will never form a blood stem cell. Finally, we have the most specific type of stem cells, which are our unipotent stem cells, and these can only generate one type of cell. An example is your muscle stem cells. They can divide and create a new stem cell or just a muscle stem cell. I really like this image here because I think it's representative of the hierarchy that we can find in stem cells. So a pluripotent stem cell will be at the base of the stem cell tree. It can still go up and divide into the main germ layers, and then specify, kind of moving towards the leaves of the tree, specifying to a specific type of cell. Now, before I move forward, as I mentioned earlier, multipotent stem cells are found in adult organisms, but pluripotent stem cells are the type of cells that we would only find in embryonic tissue. And it will be important when we move on and talk about induced pluripotent stem cells. This is the first engineering aspect that we're going to look at today. Induced pluripotent stem cells are an artificial type of stem cell. So they are pluripotent stem cells, so they can divide into any type of cell, but they are generated from adult differentiated cells that are engineered, that are treated, and they are reprogrammed to and pushed back to a less differentiated state. So we start from an adult differentiated cell, such as a skin cell, and through engineering techniques, which we can go about in more detail later, we push them back and create a stem cell. So you might wonder, why go through this process and generate and engineer stem cells in the first place? Well, being able to use to generate stem cells in the lab, it allows us to not have to actually take the actual cells from the embryonic tissue, which has its own set of ethical implications per se. And having access to this type of model can also allow a whole set of research experiments and a whole set of investigations that we would not be able to do just retrieving cells from the embryonic tissue. And finally, we can also engineer cells so that, for example, they will glow, or they will express they will express something that we're looking at. I really like this diagram that I reported here because it demonstrates the steps, what steps we have to go through to generate the type of pluripotent stem cells that we use in the lab. And it's actually something that it has been done in this lab before, and it's something that we rely on every day here. So we have done this to generate disease models, particularly to generate Huntington's, Alzheimer's, and ALS cells. So the first step would be retrieving cells from a patient. In this case, why this is very useful is because Huntington's and Alzheimer's disease, they are disease of older age. And so you cannot retrieve a sample from an embryo unless you already know the disease is there. It's definitely easier and more accessible, and there's more ethical to retrieve this kind of retrieve samples from a patient, a consented patient. So we can do this through doing a skin biopsy, and the cells that we're gonna retrieve at this point are differentiated cells. So they're differentiated cells, just simply cells that you would find on your skin. Then we go through a nuclear reprogramming, in which the cells get transfected with a virus, and a set of genes get turned on, and we achieve pluripotent cells. Now, these type of cells we can grow in the laboratory, we expand them, and then we can, through a set of directed differentiation, we can create the cell types that we want to look at. For example, neuronal cell type, like in the case of neurological disease models. We actually do this in the lab pretty frequently. We have made neuronal models starting from both breast cells as well as skin cells. It's important to note that this process is it is complicated. It requires a lot of time, and it requires a lot of dedication. It's really easy for these cells to differentiate the way and go kind of the way they want to go. So I have reported a picture here of the actual our actual iPS-derived neural cells look like. These here are neurons that were generated starting from cells that were retrieved from breast tissue. They were reprogrammed, we created iPS, and then the cells underwent a differentiation that pushed them towards neural stem cells, a multiple potent stem cell line, and then we pushed them further a little more, and we generated neurons. And in this case, the image is just it presents this type of staining with these colors because we were doing a proof of concept that what we're looking at and what we created are actually neurons. So you'll see that this red staining, we stain to target a marker that is specifically for neurons, and then this green, it stained the tubular protein. Now, what can happen if something doesn't necessarily go wrong, but what happens if the culture is not as precise as it's supposed to be, is that the cells sometimes will go and differentiate and kind of do what they want. And it's happened to us before. You can see the video here. If I can start it, okay, you'll be able to see, this is our stem cell line, our iPS culture, induced pluripotent stem cell culture. And if you observe closely in this video, you can see that the colony, these are our stem cells, actually started beating because they differentiated spontaneously into cardiac cells. I'm going to show you one more time. It's really interesting. This was not meant to happen, but it was still a very interesting experiment, very interesting thing to see. So you can see this part of the colony is actually beating on its own. And this is to demonstrate that these cells will actually differentiate into different various cell types. Now I am going to let Mackenzie come in and show you one practical application for our neuronal lines that we have generated. So one thing that we use this neural lines for is looking at Huntington and Alzheimer's diseases and possible treatments. And one of our instruments is actually able to detect the spontaneous activity of the cells. So, okay, let me turn my video off. Okay. It looks like we're having a little bit of issues to connect from our phones, but if we can get that working, we can hop back and show this again. It's really interesting to see. But very briefly, we can plate these cells on specialized plates that have electrodes, and these cells are actually able to fire and communicate to each other and work like actual functioning neurons, which is really amazing if you think about the fact that we started from a cell coming from from breast tissue, went all the way back to stem cell, generating neurons, and these neurons are actually functioning cells. So the second major aspect of our laboratory, like we introduced earlier, is the generation of tissue-derived substrate. What does this mean? So whenever if you think of cells in your body, the different cells in different areas in your body are going to have their own specific environment. When we study cells in the laboratory, most of the times they are in a artificial environment on a petri dish, which is plastic or glass, but nevertheless is very far from your actual tissues. So the laboratory techniques in general don't often reflect the true conditions. So even though we do the best we can to get as close as we can to what really happens in your body and have as close of a picture as we want, it's not a true reflection of what goes on. And there are a lot of studies that show that the actual the properties of the cells are affected to some extent by their disposition, for example, whether they are in 2 dimensions or in 3 dimensions, because that affects the way they can be in contact with other cells, the way in which they're in contact with whatever other surface they're found on. So the substrate and their environment in general does affect the cell's behavior. And their environment, it can mean, for example, whether their substrate has is hard versus soft, whether the substrate has some specific physical properties, or whether the cells in an environment are subjected to external forces to a certain extent. So what we try to do in the lab is generate a substrate that is a little more biomimetic. And what this means is a substrate that can get us one step closer to reproducing the conditions that we find in the human organism. And one way that we do that specifically is by looking at the extracellular matrix. So very briefly, the extracellular matrix, or ECM, I'll refer to it as that, is that set of proteins that is found outside the actual cells in your tissues. Is a set of proteins that is produced by the tissue themselves, and it is variable across the different organisms. So the proteins that are found, the extracellular matrix that will be found, for example, in your heart, is very different from what you will find in your brain, because you have different cells in your heart versus your brain, and they need a different type of support. For example, the heart cells, they move, they beat all day long for all your life. So their external structure that supports them need to be much stronger as well as flexible than the structure that needs to support your brain cells. Now, the ECM is also very important because it allows and it promotes communication between cells, and it allows for promotes cell-to-cell signaling as well as cell-to-cell, cell-to-ECM signaling. And this communication, the transmission of signals, is shown to it plays a role in tissue development and tissue behavior. So what we do in the lab, a little more specifically, is we create substrate derived from the extracellular matrix of various tissues. We do so because in this way we can create something that is not artificial, is specific to a tissue. So, for example, we create a substrate that is derived from the nervous system or from a brain, and we use it to grow and to look at neural cells. that brings us, it's a little more representative of the real environment of the brain compared to whether we use a synthetic substrate to grow the cells onto. This also allows us to study and look at the effects of the actual substrate of the ECM on the environment and on cell behavior. So we very briefly look at two major things in on a daily basis. We do use breast tissue derived ECM, and it has been used in our lab to look at the behavior of breast cancer cells. And then we do use brain tissue derived ECM. So very briefly, breast tissue derived DCM, I just reported a nice picture here to show that we were able to extract the extracellular matrix from human breast tissue, and through a series of chemical and treatments, create a hydrogel in which the cells can be grown into 2-dimension and 3 dimensions. Here in the image you can see breast cancer cells as well as normal type breast cells that were used for an experiment, and they were able to grow and into the substrate. A little more specific, I'll go a little more in depth into our brain-derived DCMs, just because that's what I've been doing for the last few years, and it's the focus of my research. So our brain-derived DCM, we extract the proteins from porcine brains, and through a series of steps, like I mentioned, we achieve the formation of hydrogels that can be used for growing cells into and on top of. So very briefly, you can see here we start from fresh brain. We extract it directly from the animals that have been already sacrificed for they come either from a butcher shop or they've been used for other experiments. We made it a point to never just to sacrifice the animals just for the brains. We then decellularize the tissue, and the way we do this is we've cut up the tissue into smaller sections to allow for more surface area to expose it a little bit more to the actual solvent, and then we had the tissue incubate inside of a detergent, and what this does is it blinds the cells, it destroyed the cells, but it did not impact the actual proteins. So if you can see here in the second picture, this white substance is really the brain proteins after the treatment. When it comes to the actual brain, like I mentioned earlier, there's not a lot of protein content compared to the cellular content, so we lose a lot of volume when we go through this first step. What happens next is we lyophilize the tissue, and we make it into a powder. We then digest the protein so that using a pepsin solution, so that this goes from a powder to a solution, a liquid, and then we neutralize the solution. And at this point, we have a product that when it gets put at 37 degrees, so the normal body temperature, and it's what the temperature at which we normally grow our cells in the lab, it creates a gel. So this gel gets used for further applications. We have used it, for example, to study how the cells respond to being grown in 2 dimensions on this different substrate, on this substrate. We've been able to show that the cells can grow, they can proliferate, they survive, and they can also differentiate. So in these 2 pictures that you can see, these are neural stem cells. Once again, they've been grown on brain-derived brain-derived hydrogels rather than commercially available products. So the protein that they were in contact with are brain-specific proteins, and we were able to show that through staining, they're still neural stem cells, and they can also differentiate. So the staining in this first picture in the red is a marker specifically for neural stem cells, But then in the second picture, we stain for one more time in red. If you can see it's it's faint in this image, but if you look at the red, it's neuron specific marker, and the green is a tubular marker. So I'm getting a signal from Mackenzie that she was able to get the video working on her phone. So I'm gonna go back just very briefly to what we were talking earlier. I'm gonna turn my video off and we're gonna show you our neurons that are derived from stem cell derived neurons that are firing spontaneously. Let me turn my video off.
Oh, is it not letting me use the video now? Oh, it still has you. Hold on. How does it make me the speaker?
You are the speaker. So you just have to share the screen at the bottom, and then it should allow you to show.
The camera is on. Oh, I am? Okay. Well, it doesn't show up on here. So. Okay. Just switch now. Okay. So now that everything's working, this right here is our Axion Maestro. It is a microelectroarray. Basically, the function of this machine is to monitor the electrical pulses within neurons and also the beating present within cardio or heart cells. Okay, there it goes. Now it shows up on my phone. So these specialized plates actually have a little array of little metal dots that pick up electrical signals. And they're arranged in this nice little well. So what we do is we plate cells within that tiny little dot right there. And then we let them grow. And then when we are ready to take a recording, we put it in this machine. And then this machine picks up signals on each one of those tiny little metal dots, each one being its own sensor, and brings it into our software. So right here, I'm currently only monitoring these three wells because those are the only ones with cells in them. But it'll give me a spike detection. Give it a second. They're not very active at the moment because I disturbed them earlier. They're a little finicky. So when these spike, it has the live feed of each one of those electrodes and the electrical pulses currently running through them. And then you can switch between each well. So there's the one. So then not only the live feed of the electrical pulses, it will track the waveforms of those electrical pulses and make it so that we can kind of visualize the electrical activity occurring within neurons. Since these are not very active at the moment, I'm going to go open one from yesterday that I took. Sorry, it's the live play. This one. Sorry, I'm still learning software. Wrong button. Nope, it did not give me everything I needed. There we go. Okay. This is a recording that I took the other day of the neurons we have on the plate when they were more active than they are currently. As I said before, they are kind of finicky and like to be undisturbed because, you know, axons and dendrites are incredibly delicate. If you break the connection between neurons, they don't like to communicate and they can't actually carry out their action potentials. So with that being said, this is our most active well, and it will give me the waveforms of each of the electrical pulses that this individual electrode is picking up. That makes a nice little map. And this bar here kind of adjusts itself to the intensity of each of the electrical pulses that are currently going through that given well. And then once we have this data, to record this data, and then we input it into this little metric we have, and it'll give us an actual readout of, you know, our firing rates, the the bursting patterns, as well as like the number of bursts, and other features of the electrical activity that it's happening within the cell. So it actually gives us a lot of information in regards to how healthy our neurons are. And it gives us a kind of a baseline of how we can compare our healthy neurons to our model neurons for Alzheimer's, Huntington's, and ALS, and also give us kind of an understanding of how the treatments we're applying are benefiting these cell lines and kind of reverting them back to a more healthy state. So it's a pretty nifty machine. Yeah, you can sit here and watch it all day. This is all it pretty much does. So.
All right, thank you, Mackenzie. You should be able to see me again. I'm gonna start sharing my screen again. Okay, so starting again from where we left off. Again, our brain-derived substrates, we have shown that we can grow cells in 2 dimensions, and this image that you're seeing right now is just a proof of concept that we can grow cells in 3D conditions as well. I would like to move on though and show, talk a little bit about the actual biomedical instrumentation that we rely on every day. It's not a great picture, but so one of the instruments that we use, and that was actually fundamental to get some of the pictures that you were just able to see, is our 3D bioprinter. All this machine is a commercially available 3D printer, an extrusion-based printer. It's a Fuchs model specifically, and this was readapted by a PhD student that was in this lab before us, and it was adapted to be able to be used for biological for the purposes of this lab. So what it does is, this piece that you see here, this piece that you see here, we have we can fix a needle. I can pull up a image. The needle will look like this, and the needle tip on the printer, you can actually see it a little better on this image. And what we use those needles for is we will put our cell solution within the needle, and then we use a coordinate-based system to control the actual printer, just like a regular 3D printer. And we can inject a certain number of cells in a controlled manner within our substrate. So for example, when we do our 3D culture, we can dispense our cells into either our commercially bought substrates, or into our tissue-derived substrates, like our breast-derived matrices or brain-derived matrices. We can grow the cell type that we want to look at, and inject a certain amount into a certain spot that we predetermined. I'm going to show you an example here. So this is a proof of concept for what I just talked about. So the use of our 3D bioprinter. This image comes from a paper concerning the actual adaptation of the 3D printer for biological purposes. And what they did is printing breast epithelial cells into a 3-dimensional substrate. And as you can see here, the printing pattern is very defined because it was done programmed in the 3D printer, so that this certain number of cells, which you can see is pretty consistent among the injection sites, was done at a certain distance from each other and at a specific location within our 3D substrate. And then using this instrument, we can still grow our cells throughout a period of time. So as you can see, these cells survive for over 10 days, and they've shown that the cells can grow, and we can study the behavior on the cells throughout in specific conditions, and more importantly, in 3-dimensional conditions, because if you remember what I mentioned earlier, even the 3-dimensional and actual spatial disposition of the cells, it plays a big role into their actual physiology. So what you're looking at here is the fact that printing cells using the 3D bioprinter in a 3-dimensional environment and in a way that they were distributed at a certain distance from each other. After a couple of weeks of incubation, these cells were able to generate the natural structures that they would form in the body, which is the structure that you would see in the mammary gland. And you can see here, this is a nuclear staining. So everything that is stained in blue are cellular nuclei. And so these cells, starting from getting printed, individually, they were able to grow, connect, and form a duct structure that you normally would see in a healthy tissue. A second adaptation of the 3D bioprinter that we use in the lab is to connect it and and use it as a picosecond pulsing device. So this device also, again, takes advantage of the 3D printer structure, but instead of having a head where it's connected to a needle, it's in this case connected to an electrode. So it's this image you can see here. We have these tiny little electrodes. It's connected to a pulsar, and once we activate the device, actually I can show you the actual probe looks like. So this gets connected to the printer. We have the electrode. They're very small, so I doubt you'll be able to actually see them. But we can fabricate this in the lab. We make the heads ourselves. And what this does, we can deliver an electric pulse to the cells, again, in a controlled manner. We can create a certain pattern that we want to look at. And here in the image C, you can see this is a representation of one of the patterns that we can deliver the pulses or do the prints at using our the 3D printing software. So in this case, this is a summary of one of the experiments that we've done in the past in the lab. So we use our pulsar to deliver picosecond pulses to neural stem cells in this pattern and within the cell culture well, so the normal conditions of the cells. So what we've been able to see in this case is that delivering a certain pulse does have an effect, at least on neural stem cells. What you're looking at here is a picture of neural stem cells that were treated with picosecond pulses. And this first image here is the most, these first two pictures are the most interesting, because what you're seeing here, again, this is a nuclear staining, the same that we were looking at previously. And as you can see, there's cells in this whole area. Now, what we're seeing in this first image is these green cells are cells that were able to divide after being treated with the pulse. So the pulse was actually delivered only within this area. So what we were able to conclude from this first experiment is that there is an effect on the cells, and specifically, these cells were not able to divide after the treatment. It was really interesting because this field of picosecond pulsing and using these pulses is not actually, there's not actually a lot of research on it. So just an interesting piece of data. And one practical application, one regenerative medicine application that we do use in the lab of picosecond pulses is actually trying to target Huntington disease cells and seeing if we can affect the intracellular protein aggregates that characterize the cells. So this is my last slide, and what you can see here, these are neural stem cells. They are Huntington line, and this first picture is untreated cells. So what happens in Huntington disease is there's a mutation that leads to the presence of a formation of mutated protein. So we're going to have mutant Huntington rather than Huntington. And in this picture in red, you'll see the normal form of the protein, which is it's still there even in disease condition. And then in green, you would see the mutated protein. Now, before treatment, you don't see a lot of green staining because the proteins tend to form clumps. So they're very localized just here where the arrows are pointing. Now, after treatment with picosecond pulses, you can see that the staining was distributed across the whole cellular body. And this, it was indicative that probably treating with the picosecond pulses had an effect on the actual protein aggregate structure. So this is one of the things that we're currently looking at right now, and one of the focuses of our laboratory. And it's our ongoing research. So, like I mentioned, this was my last slide. To recap a little bit, we do use 3D bioprinters and picosecond pulses, as well as we do tissue engineering of different animal-derived and human-derived tissues to create specific substrates for cell culture. We do engineering of stem cells to generate disease models, to be able to test the application of picosecond pulses, or just the effect of our 3D environments on disease and non-disease cells. That was all that we have. That was all from us, unless Mackenzie or Caleb want to add something more.
Well, really quickly, because I know we only have about a minute before Q&A, I do have these images of liver that we're currently working on, a porcine liver I picked up from a local butcher shop. So here is a photo of it. It's currently in a N-lauryl sarcosinate sodium solution, and as you can see from the outside in, it will start, it's a detergent solution, so from the outside in, it will start to remove cells, and you're starting to see this white connective tissue. So hopefully we can have some future applications, maybe hydrogel or 3D printing organs or anything really. And then there's a video of it shaking. So it's pretty neat. So that's what it does for about 24 hours at a time. And yeah, that's what we're working on next. So anyways, that's all for me.
So again, if you remember, what he's showing is the first step of the decellularization. So that's a new tissue. I didn't talk about that a lot during my presentation, just because it's a new aspect of the research we're looking into, but it's a great example of the decel process.
That was really great. Thank you so much for sharing your knowledge with us today. It's honestly one of the most fascinating presentations I have seen. And I mean, it just really seems to reveal the technology behind what you're doing and all of the science behind it, and the fact that you will be changing lives in the future. So thanks again for all of the hours you spend in the lab and in your work to make it possible to make enhancements in health science field in the very near future. So thanks again for your continued innovation. And while we continue our conversation with our speakers, I want to remind our audience that you may now continue to submit your questions in the Q&A box at the bottom of your screen, and we will answer as many as time allows. And so one of the questions that we received is, someone said that they read an article that the various kinds of epithelial, I hope I'm pronouncing that correctly, cells can be modeled by amplifying the physics of foam. Is this a relevant detail in how your research is conducted?
It is, to an extent. It does touch on what we were talking about earlier, on how the physical structure of the substrate and the external forces that our cells could be subjected to affect cells' behavior, or even just the cell like shape and morphology. So it does touch to an extent. It's not one of the main research interests that we focus in the lab. So our lab doesn't really focus on studying on how the actual 3D cell disposition or how the forces will actually affect the cells, but it is definitely a factor that plays into cell behavior. So yeah, that is correct, if I'm reading the physics of foam correctly.
Thank you for that. And what are some of the most common questions that you get when you tell people the type of work that you do? Because I imagine it's not very easy to explain to people what it is that you do.
It is really hard to explain it, especially quickly. Well, one of the questions is, people are often really interested in our disease models, and we certainly are. It's really amazing what new technologies can do. So what we get asked is obviously like, if we can, are you curing Alzheimer's and Huntington's? And like that's a big, it's a long way to that. What we do here is looking at a little something that could possibly be done, a way that we could possibly go down and possibly help. It's a lot of question marks, but you always have to remember that the research process is very long and complicated, and every new and exciting thing that we do is it's a little step.
Did you ever imagine that with the technological advances, what you're doing would be possible? It just seems really sci-fi to me when I hear about your work.
Honestly, it's one of those things, I didn't until I got in this lab and I experienced it firsthand. I still get really amazed about what we can actually achieve, especially considering that these technologies have not been available for that long. And honestly, today was the first time for me to also see that we were able to actually get neurons firing.
Wow.
So that still amazes me too.
That's really wonderful. And Caleb and Mackenzie, if you're able to turn on your cameras and want to join the discussion as well, please do. I know that we have so many aspiring scientists joining us, and they would love to know more, about the work that you do. And also, like, can you answer that question in terms of what are some of the most frequently asked questions that you get, and how do you answer them? I'll start with you, Caleb.
Yeah, the one I typically get is how will the research apply to, like, what's the point of the research, or how will it help me or others? I've talked to a lot of people that have maybe like end-stage renal failure. I actually talked to an Uber driver who had end-stage renal failure, and he was talking about like, oh man, maybe you can, regenerate a kidney or heal a kidney or whatever, for the future, stuff like that. So I always get very curious, like, wow, could you regrow organs, or can you, bank stem cells, stuff like that. And people are just generally very curious, and it is kind of hard to explain sometimes, or at least try to put it in terms that, the common, the layman can understand, so.
Thanks for sharing that. And Mackenzie, what about you?
One of the biggest questions I get is similar to Martina's, in that, where do you see this research going in the future? And are you going to cure Alzheimer's? And, I'm susceptible to Alzheimer's because my mom and my dad both had Alzheimer's. And would your research help me not go down the same route they did? And my answer is usually, I don't know. Because there's so many factors that contribute to the development of Alzheimer's. But the research that we do is kind of just a small piece of a widely researched topic. So we are just contributing to the pool of knowledge that will hopefully be utilized for new therapeutics and pharmaceuticals.
And one question I have is, what inspired you to go into the field? And I'll start with you, Mackenzie, because I think you kind of hinted, perhaps, I don't know if it had anything to do with your parents, but did it? What inspired you to go into this field?
So my background is a little bit more different. I actually originally wanted to go into infectious diseases. So I entered the program with the intention of doing that because I really like viruses and bacteria and stuff like that. But I actually ended up leaving that lab that I was working in at the time, and just kind of came here on the basis of I need a new lab and I want to stay in the program, and then just kind of fell in love with the research.
So it just speaks of, to our students joining us today, just the opportunities that await you when you go to college. You can always, even when you're in high school, it's never too late. You might be able to, talk to some researchers about the work that they do, and you may run into a lab that actually is doing some really innovative work, and something that you find that you definitely love and have a passion for as a scientist here have done today. And Caleb, can you talk about what inspired you to go into this field?
Yes. So from a young age, I've always been interested in this whole concept of stem cells, but what really drove me to pursue regenerative medicine and tissue engineering in general was when I started working at an organ procurement organization, and I started to see the effect that the research was having on donor recipients and donors in general, and the gift that donors are able to give to people and change their lives, and that really had an impact on me. So I worked with bone allografts, and that helped reduce bone, enhance bone healing rather. And it really it really drove my passion to see, these people and how they, their story and how they were affected. And it was really amazing to me. And I hope maybe one day research that I do, or, my colleagues do, can give that to somebody, Yeah.
Sounds like you're already on your way to that. Thanks for sharing that. And Martina, what about you?
I've always been interested in studying and learning about neurological diseases. There's still a lot that we don't know. There is just a lot of times it seems it's one of the, I guess it's always been really scary to think about for me. So I've always wanted to learn about these things and know more. And then I kind of happened into this lab, by accident, because I'm technically in biomedical engineering program, but my background is a lot more, it's biology. And at the time we were doing, a neuron-based project. So I kind of got directed here, and then I learned about, the aspect of regenerative medicine and translational sciences. So being able to do research with the goal of it being applied in a clinical setting. And I really love the fact that, what we do here, the goal of our research is, look into something that can actually, help with the goal to help people that could make a difference into, these neurological diseases, so that it's not something that, we just learn about and we're hoping to never have to deal with, but we can actually start making a difference. Thanks
for that. Really inspiring stories. And we just have time for one last question, and that is coming from Austin Shipley. And he would like to know, since beta amyloid plaques is one of the manifestations of the Alzheimer's in the brain, are you trying to reproduce these conditions for analysis?
Yes, that's a great question. Actually, yes, we're still at the early stages of looking at this. We're very much trying to actually get our systems to work consistently. And I think that it's one of the things that even when as students we get in the lab, we don't realize how much time and effort it goes into like the setup stages to get things to work properly and consistently. But yes, one of the goals of using Alzheimer models, other than and not just Huntington models, is trying to target the protein aggregates that are characteristic of Alzheimer's. And yes, we would want to try and have three-dimensional models to at best reproduce these conditions.
All right. Well, thank you again. And on behalf of the REYES program team, I want to thank you again for joining us today. We will continue to follow your innovative work wherever it takes you. And of course, in a future where we might, people might have a longer life because of the work that you do. So thank you again. So we're going to continue again to follow your work for years to come. And to our audience, thank you again for tuning in. Today's session has been recorded, and if you'd like to watch it again, you can share it with friends on our YouTube channel, which is linked from the REYES website at odu.edu/REYES. This concludes today's session, but I do want to remind you that we will continue our Python for STEM class next week, so please tune in, look at the REYES calendar, and be sure to continue following us so that you can learn some more coding on Python. So please have a wonderful weekend. We look forward to seeing you again next week, and have a wonderful day. Bye.
Thank you for having us.
Thank you for being here. Bye, everyone.